up in Arkansas. My father was actually in state government, so we lived in and around Little Rock most of the time. But the family farm is in the very southwestern corner of the state, so home is where the farm is. So that really was where I spent a heck of a lot of my time.
Tell me about the farm.
Well, at that point, we were raising cattle primarily, and a lot of timber. And when my uh, uncle, who stayed on the farm and really was the one driving the cattle production, when he got to where he could not handle cattle anymore, we went to straight timber. So, and that's what it is today. The neat part about it is now that I'm back in Arkansas with a faculty position, the family farm is also my field site. Really, I was always very interested in science, so I knew that my career was going to be something in the science arena. Exactly what I didn't know. In high school, I had interest in veterinary medicine. I also had interest in human medicine. And then when I started college and taking the courses, I really was enrolled as pre-med until I took a course called Natural History of the Vertebrates. And that is the one that totally changed my direction in life. After taking that course, now it's called vertebrate zoology, but after taking that course, it was very clear that I wanted to do work with wild animals, and I wanted to be a university professor. The great thing about that course is it was taught and still is taught with a very heavy field emphasis. So the students learn all of these animals by handling these animals. We get all of the students out in the field. They're catching salamanders and fish of various kinds. So the students learn to seine fish. They learn to collect snakes, collect lizards, collect small mammals. It really is almost 100% in the field. Yes, there's 2 hours of lecture each week, but there's also 4 hours of lab. And the way I teach it is I have two required field trips. One of them is a weekend field trip, so we leave on usually a Thursday and come back on Sunday. And the other one or two are nighttime field trips, one mist netting bats, and then one where we do powder tracking with small rodents. So that course, doing that kind of work, that convinced me that medicine was not for me, that it was going to be field biology. My undergraduate advisor, Gary Height, got so many people interested in field work. Many of his students have gone on to be professors at other universities. Some of them I count as colleagues still today, and we interact. Yeah, Gary was a big influence on me.
What was it about him that was so influential?
That he just enjoyed getting students out doing this and just really having fun with the students in the field. Um, he just made it a fun class. One thing that really did play a huge role, quite frankly, is the support I got from my family and from my wife after we got married. So we started dating before I got into this field biology business. As a matter of fact, we were um, college students together, and she was completely supportive. She did draw the line. We had to have his and her refrigerators and freezers because she was not going to allow my animals in her freezer. But beyond that, no big problem. My parents also completely supportive. They thought it was a little strange, but okay, that's fine.
What did they think was strange? What piece of it?
Well, when my mother came home and she was rummaging through her freezer and pulled out a Ziploc baggie, a pickle. Who froze a pickle? Why would someone put a pickle in the freezer? No, Mom, it's a shrew. What's a shrew? So, yeah, I was pretty steady at putting animals in the freezer.
And yet they really appreciated your love for
Oh,
this.
absolutely. Absolutely. And they still do. Both my parents are still living and they're getting a kick out of watching this happen. So, and they're still completely supportive. And the family farm still is my field site. As a matter of fact, the, my extended family, my grandparents played a pretty major role as well in the whole education bit. My grandfather never had an opportunity to graduate from college. He did take some college classes, but didn't graduate from college. My grandmother, on the paternal side did graduate from college and in fact had a nursing degree and was a nurse. Um, but my grandparents thought that education was so important that for high school graduation, they gave each of their grandchildren a college education. So that really kind of pushed us in that direction. I knew that graduate school was my next step. I started taking every field course that I could at the university. Um, so I followed the vertebrate zoo course with mammalogy, and I eventually became a mammalogist. Um, I took ichthyology, and then we just simply had a lot of field work going on. The university did not have a graduate program at that time, but the faculty members were still very much involved in research, so they used undergraduates. That kept getting the undergraduates out in the field as well. We actually started some armadillo work, again, down on the family farm, um, when I was an undergraduate, and then we just kept rolling with it through graduate school. And that armadillo work, at least a portion of it, became the first paper that I ever published as a scientist. Um, gosh, I was in my master's program at the time, but it grew out of that work that started as an undergraduate.
When you say armadillo work, what is it that you're studying?
Well, for the armadillos, we started off, and this was to be my master's project. This is what we're planning on for a master's project, would be looking at home range and habitat use of armadillos. So we needed to put radio transmitters on them. Remember, this was a pretty small school at the time. We didn't have funding for this research, so I had to build my own transmitters. And I disassembled some. I learned the schematics, and I learned how to make radio transmitters to go on these animals. Making the transmitters was not that difficult. Had a product that would really work well, had good range. We even built the receiving antennas, so all of that worked well. The problem with radio transmitters and armadillos is attaching them. You can't put them on their neck because their shell prevents that. I could not make them small enough to put the transmitters inside the animal, a surgical implant in the abdominal cavity. So we tried lots of different ways of attaching the transmitter on the outside. And after working for about 2 years on it, I came to the conclusion that we could not do radiotelemetry on armadillos with the equipment that we had available to us. They've got to go internal. That was what we started working on armadillos. But along with that, we were also looking at diet of these animals. So there was some, if you look at the literature on armadillo diet, some people claim they eat a lot of eggs, especially of ground nesting birds like quail. And there was speculation as to what they eat. The food habits papers that we found from different parts of their range, every single one of them differed. So we decided, well, okay, let's find out what they're eating in this part of their range, since we have this population that's very easy to work with. So we started collecting lots of armadillos, removing their stomachs, obviously, for food habits analysis, and then picking through bits and pieces and parts of crunched up insects to identify what they were eating. For my master's work, I did some systematics research. I was looking at bobcats, and really it was always skulls, looking at morphological variation in the species across the eastern half of their range, and correlations with geography, environmental factors, etc. But then what really set my direction in terms of a research direction was a paper that I read when I was still an undergraduate, on how mothers should allocate their resources among their offspring when they don't have enough resources to rear all of their offspring. So a female mammal may be carrying a litter of X number of young, certain number of males, certain number of females, but over the period of gestation, environmental conditions may change. So if she's faced with a shortfall in resources to be able to rear those young, how does she allocate those resources? There was a theoretical paper that came out in the early 80s by Trivers and Willard. I think it was 81, Robert Trivers and Dan Willard, that predicted that females should bias their investment to favor female offspring at the expense of males. There was a follow-up paper done on that where a scientist did an empirical test, and she food restricted nursing wood rats in the laboratory, and son of a gun, males took it on the chin, but the female offspring, a greater percentage of them survived. So I wanted to follow up with that work and really kind of tease out some of the details on the Trevor-Woodard hypothesis, and that became the focus of my dissertation. More specifically, maternal investment. Part of it was also how much it costs to rear different numbers of young, so the energetic cost of litter size. After I finished up with that, that became the focus of everything that I wanted to do. I wanted to look at energetic cost of reproduction. So I designed a study that just needed funding, and I fought for the next several years to try to get funding for the study. Finally did. But along the way, I had an opportunity to go to the Patagonian region of Argentina and Chile for 2 expeditions where they were really alpha level surveys, what mammals are here. So our research crew, our research team was traveling literally from the East Coast to the West Coast and back, or West Coast to East Coast and back on each of these expeditions, collecting every small mammal that we could and trying to identify what species were there. We were also looking at patterns of gene flow from when that region was glaciated. That got me pretty hooked on that type of field work. 6 weeks in the field, it's a great way to spend some time. I was following up on Polly McClure's work where she demonstrated this sex-biased maternal investment by wood rats, and I was food restricting these mothers. And when I tried to publish that paper for the first time, it went to Animal Behavior, and when the reviewers were going through the paper, if any reviewer has any question at all about the care of the animals, they can flag it for review by the internal Animal Care and Use Committee. Well, mine was flagged because I was food restricting these animals and, in fact, inducing infanticide, which is exactly what these mothers would face in nature. And when I found out that, okay, they're going to review my paper, I thought, well, gee, if people are going to be making these types of decisions, we need to have people on those committees that really understand what's going on in nature with these animals. So I immediately, or very shortly after that, tried to get on the Animal Care and Use Committee for Animal Behavior Society and also for the American Society of Mammalogists, which is my home society, and served on both of those committees and still serve on the Animal Care and Use Committee for the ASM. These are the Animal Care and Use Committees of these professional societies. So it's not an institutional Animal Care and Use Committee. These are the committees that establish the care and use guidelines for that professional society. We had to sort through over and over how you fit the requirements for wild animals into the guidelines that the institutional Animal Care and Use Committees expect you to adhere to. And the fit is not smooth, as one would expect. This is going to be a never-ending process. In fact, one of the sessions that I'm doing at this meeting is focusing on one of those things that, gee, no one has even addressed this yet. We need to look at this. It involves how frequently you observe animals. If you look at the expectations, and what's written in the Animal Welfare Act and the Guide for Care and Use of Laboratory Animals, it's, of course, check the animals every day. And in some cases, you may need to check them more than once a day. That doesn't work for some animals. If you have a hibernating animal and you check it once a day, it's not going to live very long. I mean, think about doing this with bats. If you're disturbing the bats every day, they're not going to last very long. There are other animals that, especially animals that are coming in directly from the field, you may need to keep your distance. You may need to think of alternative ways to observe those animals. For example, you bring animals in directly from the wild. These are not domesticated animals. They are wild animals. Humans are a threat to them. They have to become habituated. They have to learn how to eat different food. They're not going out and capturing their own prey anymore. So keeping your distance from those animals, especially over that initial period, is a wise move. And it reduces the stress on the animal. When you do that, the animal's going to be happier, we're going to be happier, and the science is going to be better. When we were interacting with IACUCs, at institutional IACUCs, or individuals from those IACUCs, they really didn't understand the differences between domesticated lab animals and the wild. Remember, the Animal Care and Use Committee of the ASM is composed of mammalogists. We are field biologists. We typically don't work with domesticated animals. We are specialists on the animals in the field. So for us, this is, of course, this is the way you do it. This is the way you treat these animals. But convincing the members of IACUCs that do not regularly experience work with wild animals that this is typical, this is the norm, and you cannot do it. You can't treat these animals the way you would a white mouse or a white rat. I guess that was one of the biggest challenges. I think the 2007 guidelines were a very important change because that's when we ramped up the meat of the mammal guidelines. And they became a document then, at that point, that IACUCs really had some meat to tear into when they were working with wild animals. Bill Gannon was the chair at that time, and I was pretty heavily involved in redrafting all of this stuff simply because I'd been on the committee and had been very active, I guess, in this whole arena. And I had served on my IACUC at my institution at this point. In fact, I think I was probably facility director by the time the 2007 version came out. So Bill and I, just simply because of our professional positions, we were pretty deeply involved in oversight of animal research. But we had an entire committee backing us up. Mammalia is so broad that you can't have one or two people write guidelines. You've got to have someone who is a bat specialist. You've got to have someone who is a marine mammal specialist. You've got to have someone who is a large mammal specialist, an ungulate specialist, a large carnivore specialist, a mesocarnivore specialist. So we had an entire committee that we could pull on for details that we didn't know, and we simply turned over sections to them. Look, draft this or read what we have drafted and make corrections. You are the expert in this area. So it really was very much a team effort. Our Argentine colleague got a 2-year stint to come up and work with us in Minnesota on the animals that we had collected in Argentina. So certainly I remained up there to continue that work. During this intervening time, I did field work with other people and had a couple of projects that came out. One of them was a fish paper, and certainly I'm not a fisheries biologist, but my collaborator on this one was a graduate student. He had some great ideas, and we designed a little experiment literally on the back of a napkin one day in the lab and decided we were going to make it work. So we put this fish study together, and to this day I think it's one of the most novel studies I've done.
What were you studying?
We were looking at fish, and it was parental allocation, so it really was continuing with my interest in maternal investment. But we took a single sib ship, so a single clutch of eggs from this little fathead minnow. We were working with fathead minnows, and we worked with a single sib ship because we wanted to control genetic variability. And then we divided that sib ship up among 3 treatments. One treatment got fed 3 times a day, 7 days a week for 150 days. One treatment got fed the same amount, but on a completely unpredictable schedule. And we determined how much to feed them and when to feed them by rolling dice. So One dye would tell us whether they got 1 day, 2 day, or 3 days worth of food. The other would tell us if they got it in one feeding, two feedings, or three feedings. And then the third treatment was where the fish started out on this unpredictable feeding schedule, and then halfway through at day 75, they switched. And when they became reproductively capable and started producing eggs, we quantified egg production. And then when it was all over, we weighed the fish, dissected the fish, and measured gut length relative to body mass. And what turned out, what was so cool about it, is these animals that were fed on a completely unpredictable schedule were much smaller than the ones that were fed on a predictable schedule, but they were producing as many eggs. And when we looked at their intestines were much, much, much longer relative to body mass than the ones that were fed regularly. So we end this paper with a little quip that says, it appears that there's a tremendous latitude for phenotypic plasticity in these fish, and it takes a lot of guts to have kids when your next meal's a crapshoot, which summarized the entire paper in one sentence. One of the reviewers wrote and said, I appreciate the humor with which you end your paper, but I'm not sure that I'd have the guts to do it myself. In 2011, Ellen Paul, who was the executive director of the Ornithological Council, and I were co-conveners of a conference on oversight of wildlife that we held in Albuquerque. And our goal there was to bring together regulators, IACUC members, and investigators for an open discussion of the challenges that the IACUCs are facing, that PIs are facing, and that regulators are facing when it comes to working with wildlife. And that discussion continues today. That really was kind of a watershed event in many, many ways. It's not the first time that there was ever a conference on oversight of wildlife. There was actually an earlier one, I think in 2004 at Fairbanks, Alaska, and maybe one before that. I was involved in the one at Fairbanks as one of the speakers representing the ASM and the Animal Care and Use Society, Animal Care and Use Committee. And I guess that's part of what gave me the idea that we really need to redo this, to bring it back and meet again for a continued discussion. And we have plans to continue that. The discussion was fantastic in that IACUC members that were there, light bulbs were clicking on. And, oh, okay, we do have the latitude as a committee at my institution to make these adjustments that will ease the way for wildlife researchers, but are still completely compliant with all of the regulations that we have to adhere to. And after that committee, I mean, I can't tell you how many IACUC members at various institutions that said that changed how our committee operates when it comes to wildlife. And I've had faculty members at these institutions that have told me that it's light and day, or night and day different after that meeting as compared to before. That their IACUC really understands much better that there are different ways to remain compliant, and that they don't have to be quite so rigid in their thinking about dealing with animals when it's a non-domesticated animal. IACUCs really don't have a lot of guidance when it comes to the nuts and bolts of dealing with wildlife, and for good reason. Like I said, there's over 60,000 species. No one can write a set of guidelines that's going to cover every aspect. It's simply impossible. But you come up with the general guidelines, a general framework that works for wild animals. I get phone calls, I get emails nonstop. Look, this is a challenge that's facing our IACUC right now. Tell me if we're thinking about this right. Tell me, we don't have any expertise with this animal. Can you help us? And I may not have any expertise with that animal, but if it's a mammal, at least I know a society that I can turn to, and I can poll the members in my society and odds are we can come up with someone who has worked with that species or a closely related species. We can usually get the questions answered. If you look at even the Guide for Care and Use of Laboratory Animals, it says specifically, page 32, I think, that this is not a compendium of all things wildlife, and IACUCs are encouraged to consult experts. Okay, these guidelines are drafted by the experts. There's guidelines for birds, for fish, for reptiles and amphibians, and for mammals. So it is our responsibility from the taxon societies to keep these guidelines current and usable for these IACUCs to provide that first level of resource. So this call for the recognition of these guidelines from that Albuquerque meeting, Ellen Paul and I just sit down and said, you know, we need to formalize this request and really bring it forth so that people understand that these guidelines are out there and that they should be the go-to guidelines when the IACUC is dealing with wildlife. So we put that together. We drafted in some expertise from the other taxon societies because Ellen's certainly not a herpetologist, nor am I. So Stephen Beaupre was representing Ix and Herps at the time. He was the president of ASIH. So we put together this formal request and published it in Bioscience. From that, the NSF did come back and explicitly recognize the taxon guidelines for the work that they fund. It makes it easier for IACUCs. It makes it easier for the PIs. It gives these IACUCs, I guess, assurance that yes, this major grant-making organization recognizes these guidelines, these taxon guidelines, as appropriate references when we're dealing with these wild animals. So it simply endorses what their PIs have been telling them all this time. What we're sensing is that we're meeting a need that wasn't being met at all before. It's like we've I don't know, created a little crack in this dam, and now it's just flooding through with questions, with how do you deal with this? Okay, tell us, you know, what will provide some insight on this situation with these wild animals. We've got investigators going to a foreign country. They can't take the drugs, the pharmaceuticals with them that we would use in this country. What are their what recourse do they have in this other country? So the questions are just coming from everyone now. One of the big challenges in dealing with wild animals is that the protocols in use at all these institutions are designed for domesticated species in a laboratory. They don't ask the very basic questions that are first and foremost when you work with wildlife, like how do you get the animals? You can't order these animals. In fact, for many of these wild species, if you find someone that's willing to sell them to you, that person is a felon. You cannot legally sell these animals. So the first question is, how do you obtain your animals? That's not addressed in the Guide for the Care and Use of Laboratory Animals, for example. But that's not surprising. It's the Guide for the Care and Use of Laboratory Animals, not wild animals. It is not addressed anywhere else except the taxon guidelines. So we decided that what we needed to do was kind of overhaul the whole protocol scheme and develop a protocol that is very specific to wildlife, that asks the right questions that an IACUC should ask if they're going to do a credible review of a wildlife protocol. So we start off by asking, how are you going to obtain these animals? We ask, what permits do you need to obtain these animals? That's another difference. You don't have to have permits to work with each species. In biomedical research, they're domesticated. You do have to have permits when it comes to virtually any species of wildlife. We ask questions. If an investigator is going to go out and capture these animals, usually they might capture other animals as well. So that's a whole other topic. Non-target animals. So you've got target animals, you've got non-target animals. Non-target animals in biomedical research means that you walked into the wrong lab. Non-target animals in wildlife research means that you're setting a trap and you're hoping to work with, let's say, you're intending to work with grasshopper mice, but you're setting this trap in the Southwest and you capture grasshopper mice. You also capture kangaroo rats of various species, Paragnathus. You may capture Neotoma, whatever. Those are non-target species. We address other questions like how you assign a USDA pain or distress category to the work. If you're working with wild animals, very commonly the animals need to be sedated, not because you're inducing pain or distress, simply because you can't handle them otherwise. If you're working with a bobcat, try to do that without sedating that animal. I have worked with raccoons, and one of the tasks was to age these animals using tooth wear and also to ear tag them. You can't do that with an animal that's not sedated. So you're using these pharmaceuticals to sedate them, and many IACUCs say, oh, if you're using pharmaceuticals, it's automatically category D, which is pain or distress relieved by analgesics or anesthetics. You're not relieving pain or distress. You're simply using it as a way to handle the animal. Many wild species are captured using pharmaceuticals. For example, if you dart deer, you may be using carfentanil to capture the animal. Again, it's not to relieve pain or distress. It's how you capture the animal. So that's addressed in the protocol. We touch on considerations of the natural population. Now, IACUCs typically are not in a good situation to evaluate the effect on the population, but the organizations that issue the permits to work with those animals are. So what we say is that we encourage IACUCs to use those permits as an additional assurance that any impact that there may be on the natural population is negligible or is warranted. We discuss what's going to happen with the animals at the end. Many times it may not be wise to put animals that have been in captivity back in the wild. Sometimes study designs require that. Sometimes permits require that. So we touch on the types of considerations that need to be made if you're going to release animals. So it's a protocol that's very specific to wildlife. And when we put this thing together, we very deliberately posted it in Word, not in a protected format, because we encourage IACUCs to take it, to modify it to suit their situation. If they only need parts of it, they want to adapt it into their existing protocol, fine, go for it. We're putting it up there as a resource. It is pretty recent. It's being used more and more. Institutions may not use the entire protocol, but they certainly will pick and choose parts of it. It's pretty long. We tried to make it very inclusive, and we realized that many of those areas, a PI will simply put NA. So we encourage institutions to set it up in kind of a smart forms format, which will cut the workload, I guess, down to size for the IACUC reviewing it. We did develop a wildlife course that CITI program has put up. So it is an entire course that is wildlife specific. And it like the protocol, like the taxon guidelines, it goes through the details that students or PIs would really need to be aware of if they're working with wildlife. So it really gets into the compliance aspect of it. And it discusses training, it discusses OC health. I guess I use my classes kind of as test beds for this. the first lecture in each class in which we are going to use animals, which is every class I teach, the first lecture is about how we are going to use animals. And because I use mostly wild animals in my classes, um, they get a pretty heavy dose of the taxon specific approach to working with these animals. If you're teaching a field course where students are going to be working in the field and they're going to be exposed to wild animals, I encourage people to make their first lecture one that covers the hazards that students may face. Number one, if you put it on your syllabus and the syllabus is required for the course, that's a documentation of training of those students on things like zoonoses, on how you're going to handle the animals that you work with. If you look at the work with wildlife, the primary beneficiary of that work is the species themselves, either in terms of management information or more basic knowledge about the animal, which informs management decisions. It's not like we're using these animals as a surrogate for humans because, gee, you can't do this work on humans. No, we're using these animals because we're interested in understanding that animal, maybe how they survive in their situation. It may very well inform management decisions. So the beneficiaries really are the animals themselves. So maybe that cuts down some of the controversy, but you're still using animals, and we still kill animals as part of the studies, whether it's training for the students or whether it's part of the research. So that is part of it. Is there going to be some element of controversy? There actually is. But I will share this with you. I was teaching a field mammalogy course. And in that field mammalogy course, this was when I was at Minnesota, I did have some students who were much more anti-animal use. And they were taking this field mammalogy course. And in that course, I required a collection of animals. The students had to go out, collect animals, and prepare them as museum specimens for permanent deposition in our museum. Whether they were opposed or not, that was a requirement for the course. It was not optional. All the students did. At the end of that semester, I routinely asked the class if they felt what they thought about the value of collecting these animals, and the student who told me up front that they were not, that they were opposed to collecting animals, wrote quite an extensive comment to me and said, never get rid of the collection. That is a fundamental component of this course that makes it worthwhile. So if I can get across to a student who comes in with thinking that animal use is totally negative and switch that person's opinion, then okay, maybe they are seeing that there is utility to handling these animals, to studying these animals, including putting them up into a permanent collection. The reason I require the collection is because you never really understand the ecology and behavior of that animal so much as when you have to go out and put hands on that animal in the wild. If you can go out and find where to obtain that animal, how to capture that animal, you've learned a heck of a lot about that species. All of the knowledge that we gain on a species ultimately is going to, at least a large portion of that, is going to inform management decisions. For example, Department of Natural Resources for whatever state manage their wildlife populations. They manage white-tailed deer. Okay, they know that if they go out and collect does at certain times of the year, then they can look at the size of the fetus that these does are carrying, and they will know when that doe was bred. They will know approximately when those fawns are going to hit the ground, And from that, they can infer whether that doe was bred on the first cycle or the second cycle of estrus. That information comes about by studying deer in natural populations, but that informs management decisions. If you find out that your deer are being routinely bred, many of the deer are bred on second and third cycles, so that the fawns are hitting the ground very late and they're small going into the fall season, then that may mean that your male-female, your buck-doe ratio is really out of kilter. And you need to go back and alter that sex ratio by taking does out or by protecting the bucks so that you have a more nearly even sex ratio and the females, a larger percentage of them are bred on the first estrus cycle. So how do you get that information? You get that information by studying the species. After I became chair of the Animal Care and Use Committee, I realized that one of the big pushes we needed to make was to educate IACUCs, compliance and regulatory personnel and PIs on how one goes about working with wildlife in the regulatory framework that exists. So with that in mind, where are you going to reach these people? You're going to reach them at the IACUC training opportunities, and Primer is a huge component of that. So I contacted Joan Racklin and asked if it would be possible to attend Primer and to do a breakout session specifically on wildlife. Through the ASM, I pushed the ASM to become a member of Primer and also a member of SCA at this time. And the society did. We have a society membership, and I am sent to the meeting each year as the society representative. So the first breakout session, I was on the faculty, and the reception was very positive. And it's clear that we were providing something that was not being picked up anywhere else within Primer. So I just kept the offer in to come back anytime they would have me, and I guess I've been here every year since. And the number of sessions has certainly grown. Last year, I had an opportunity to participate in a panel discussion on oversight of wildlife, and then breakout sessions as well. And then I guess after that, they asked if I would serve on the planning committee. So maybe that's putting the fox in the hen house, but of course I'm going to try to push this wildlife stuff as well. And this meeting, we've got even more sessions, including another panel that is focused on study of wild populations where the wild species are the primary beneficiaries. And our plenary speaker is talking about really diseases in wild populations and how they impact humans. I mean, the reception from Primer has been phenomenal. I mean, absolutely phenomenal. But as I said, we've kind of chipped this little hole in the dike, and the interest is just flooding through. People are interested in this because the need is not being met elsewhere. I describe myself as a behavioral and evolutionary ecologist. I really focus on behaviors and the ecology of animals in their native environments. Okay, how an animal makes its living, how a population works in its community, how it fits into its community, how the dynamics of one population affects the dynamics of another population. So that's how I get across what I do.
If you could influence a major change in how IACUCs work without regard to the need for resources, what would you wish for? In other words, how would you finish the sentence, if only?
I guess if only they would reach out. All they've got to do is call. All they've got to do is email. If they have a question, it's a taxon that they're not familiar with, it's a capture method they're not familiar with, rather than trying to make a decision in a vacuum, resources are out there. And IACUCs actually are encouraged to tap into those resources. All it takes is a phone call. I know that my colleagues in all of the other taxon societies will be eager to provide information. They will. Because it's a challenge for us when the IACUCs don't understand. The last lab of the semester is a comparative dissection. Okay, you've got to dissect in zoology. All the zoology classes everywhere use pickled fish, pickled frogs, pickled rats that are, have latex in the arteries and veins and what have you. How boring. So we use fresh animals, snakes. I bring in mammals that I have acquired in one manner or another. It may be a roadkill. Last year, I brought in a coyote that was killed on the road. It was in good shape, so as I was driving home, I saw this thing, picked it up, put it in the truck, put it in the freezer. When we do that dissection, the students get to see that animal as an animal and look at the anatomy of that animal. They get to see that animal as a habitat because it's loaded with ticks. You open it up, it's full of heartworms. It's full of nematodes in the gut. There's tapeworms in there. You get to see that animal as a predator. You open up the stomach. There was a whole chicken in that one. Head, feathers, feet, and all. There was a fragment, enough of a fragment of an animal that we could tell that it had eaten a lizard. Different mice. If you get a couple of teeth from a mice, you can usually identify what species it had for dinner. Okay? So I like to use these animals that are not pickled, not injected with latex, and yeah, they may smell like a fresh animal. But you know what? That's what nature is. And the students get a far richer experience, I think, doing that. And they rave about that lab. I mean, that lab is absolutely their favorite. They're in for an experience in 1 month. This year already, I've picked up a gray fox, a raccoon, an opossum, and as I was driving into work the other day, I picked up a deer that had been clipped on the side of the road, put the whole thing in the back of the truck, and went in the freezer whole. So that is going to be our comparative dissection. 1 year, the mix of animals that I had was I had an opossum, I had a raccoon, I had a bobcat, and I think I had a beaver that year. Okay, so you've got a strict carnivore, the bobcat. You've got a strict herbivore, the beaver, or maybe it was a squirrel, whatever it was. We weigh all of these animals, and then you do the comparative dissection. So yeah, you get all the food habits, but also you take out that intestine, the small intestine, and stretch it out for each of these animals. Literally, we will stretch these down the table. So for the herbivore, it's really, really long relative to the mass of the animal. For the bobcat, it's really short relative to the mass of the animal. What's the take-home point? Animal matter is really easy to digest. Plant matter is really tough to digest. There's a morphology that drives that point home. If you're working with fish, same thing. You do a carp compared to a bass. One has a very long intestine, one has a very short intestine. Carnivore, herbivore. Humans have great difficulty understanding another species, especially when it's a non-domesticated species. For example, the guide even talks about the guide for care and use of laboratory animals. Are you an animal scientist, by the way? Do you know what the guide for the care and use of laboratory animals is? Okay. The guide for the care and use of laboratory animals talks about need for social contact in animals, especially for things like dogs. And it said if another dog's not available, then human contact will work as well. Not for a wild animal. Human contact is absolutely the worst thing that you could do. The more you can minimize contact with these wild animals, the less stress you're inducing. Wild animals are that much opposite. I mean, totally and completely opposite direction. Our greatest difficulty, I think, is trying to see the animal and the animal's environment through the eyes of the animal. That's where we have difficulty. Um, one of the little projects that I do in some of my classes is I take students out. We capture mice in the afternoon, and I drop these mice, these different species of mice, all captured in the same area. I drop each one of them into a bag of fluorescent powder and shake it up and turn it loose. Fluorescent orange, fluorescent green, fluorescent pink, and the animal just runs off. And then we go back and sit around and have dinner. 2 hours later, I bring the students back out to where we turn those animals loose with black lights. And they have to track each of these animals, sometimes down on hands and knees, tracking these animals. But if you capture 3 different species in exactly the same general area, certainly an area small as the size of this room, you can capture 3 different species. If you follow the trails, those 3 species are using the same habitat in completely different ways. That drives home to the students the point that what we see as a type of habitat is completely different from what the animal sees as that same type of habitat. The animal sees its habitat in a manner that is adaptive for that species. When we're working with wild animals, we need to learn to try to think outside the box, to use an expression that's way overused, but to try to see the animal use from the perspective of a wild animal, not a domesticated species.